So welcome everybody to the online workshop, Bioreactions in Flow, organized by Classy as an online workshop. Well, maybe you asked, what is Classy? Well, Classy here is a fed open EC project, and we have as the main objectives to develop microfluidic platform. And I took just some significant objectives out, also to deal with immobilization of multiple enzymes and to have cascade reactions where we do selective activation or deactivation of certain steps and develop tools to optimize those. Well, the partners in Classy are actually the University Autonoma of the Madrid, the Ben Gurion University of the Negev, ETH Zurich, University of Graz, Radboud University, the Micronit, and Accelerant. And we are very happy that today 5 companies also join in: Insight, Enzian, Enzyme, Chiral Vision, and Enzymical, to present also their point of view. Well, before we start, just some general comments. You should have been informed that this event is being recorded. And if you do not want, please write to this email. Other points, please, if you're not speaking, that's especially important for the panelists, keep the microphone muted, turn the camera off. And well, as the schedule is rather tight, I will not give a long introduction to everybody. So please refer to the CV of the speakers on the workshop homepage, there to the slides. And reminding the speakers, you have a 15-minute slots, which means 12 minutes for your talk, and then we would have 3 minutes discussion. And for the discussion, there is this Q&A tool, questions and answer function in Zoom, which you may use and type in your questions, ideally already during the talk, not when the talk is over, because then we need the questions. So type in the questions during the talk. If there are any technical issues, please use the chat function to type in, and then you will get help there. Well, my name is Wolfgang Kroutil. I'm from the University of Graz, and I welcome the speakers, Professor Wilhelm Huck from the Radboud University, Christoph Winkler, University of Graz, Elwin Vrouwe from Micronit, and David Pedrosa from Insight, Stefan Bayer and Mattia Lazzarotto from Enzian, Javier Ibanez as a group leader at Enzyme, Rob Skubart as managing director at Chiral Vision, and David Liese as COO at Enzymicals. Well, you have seen the program, and there we have first 6 presentations until we have a short break, and then the last 2 presentations plus at the end a panel discussion. So not to delay any further, I will stop my presentation here and hand over to Wilhelm to be able to share his presentations.
Thank you, Wolfgang. Okay, then pleasure to kick off this afternoon's meeting. I will not introduce myself and the group, but I will get right to the sort of topic that we are working on within the Classy project, and that is optimal experimental design strategies for optimizing immobilized enzymatic reaction networks. That's a very long title. Why are we trying to do this? Our long-term goals, in a way, are to work towards the construction of a synthetic cell, or to do cell-free biosynthesis. And in both cases, you will appreciate that we will have large enzymatic reaction networks. And of course, we can try and construct those large enzymatic reaction networks by picking carefully or choosing carefully the different enzymes. But the problem is they need to have a certain function. And function here is that they need to produce the optimum amount of yield, the optimum efficiency, cofactor consumption, maybe stable under fluctuations in input, etc., etc. And so this is going to be extremely challenging. It's challenging to make the network. It's currently almost impossible to control the output of the enzymatic reaction network because we really don't have enough data. What do I mean with that? Well, an example that we have been working on is that we work, for example, with a nucleotide salvage pathway. And in that case, you have a small enzymatic reaction network that starts with an activated sugar and then has the different enzymes that you need to connect the nucleobases to the sugars and then phosphorylate them to make the 3 sort of triphosphate nucleotides. So that looks like a fairly simple enzymatic reaction network. You have about 6 different enzymes, you have 15 different species, and you have competition for different substrates by different enzymes. And so it turns out that that is actually already an enormously challenging system, because it means that any of the or many of the rate constants that you have in this reaction network, they are coupled in such a way that you can sort of not really simulate the outcome of this reaction network, simply because your sort of outcome is depending on whatever choice you have for one parameter immediately is counterbalanced by another parameter that is sort of in communication with this parameter. So it means that to optimize the outcome of this network, I cannot simply rely on sort of solving this set of differential parameters that I get out because I don't know all the kinetic parameters yet. So the way we are going to solve this is by having this optimal experimental design pathway. And so what we will do is we will say, well, actually, we're going to immobilize the enzymes in a reactor. And so I will show you in a minute how we do this. So we have a continuous tank reactor, a flow reactor containing the enzymes immobilized on beads. And then we have 6 different inputs. We have uracil, GMP, adenine, ATP, PEP, and PRPP. And then at the output, we have 15 different outputs. So that's partially the inputs that we put in and the intermediates that are formed and the final products that are formed. And what we can use HPLC to try and analyze as much as possible what comes out of the reactor. Turns out we cannot see everything. We see about 8 observed species. And I'm going to explain to you later on how we're sort of analyzing the kinetics of the network. The first important step is that we are immobilizing the enzymes. And I'm sure there are some specialists in the audience, and we can have further discussions. What we do is we use microfluidics to make a micrometer-sized gel bead, say 20 to 50 micrometers in diameter. And this gel material can be activated using EDC coupling, and then we can couple any enzyme to these beads. There are different ways of doing this. We can also immediately sort of immobilize the enzyme while we are making the gel beads. There are different ways of doing this. The net result is that we end up with sort of very uniform beads with a certain amount of enzyme on there. And we can check for the enzyme activity, of course, before we load them all in the reactor. When we mix everything in the reactor, we have lots of different beads of lots of different enzymes, so the whole network is reconstituted, but it doesn't flush out. So the sort of small molecules go in and out, the enzyme stay inside. So now, how do we obtain a maximum amount of information from experiments like this? Well, the way to do that is by having an algorithm that says, well, rather than flushing in a certain concentration of substrate and look what the conversion is in your reactor, we are going to continuously vary the input concentrations. We're also going to change the flow rate over the course of the experiments. And then we continuously monitor, or we sample the output of the reactor, and we're going to measure the concentration of all the different compounds. So that is our informative database. So with a certain modulation of the input, we get a modulation of the different species that we measure. Then what we do is we try and use our kinetic model to fit the sort of based on this particular input pattern, this particular output pattern. And in the beginning, that doesn't work so well. So your predictive power will not be so great. But once you have done this once, your algorithm can now suggest a new modulation for input variations that we can then use in a second cycle, and then you get much more data that eventually sort of allows you to fit your data in a much more accurate way. And so this is an example. The algorithm suggests a certain input modulation of the sort of different inputs that we have for a reactor. We run this for about 8 hours because these reactions are not super fast. Then you see here that these are all data points that we take from our HPLC. So we measure hundreds of samples, definitely over the course of many experiments. So this is about 70 samples that we measured, where we sort of determined the concentration of the 8 output species that we can detect accurately. And then we fit this data to the model. So the model would predict the solid lines of output species based on this input concentration, and the dots are how close we are to that prediction. Now, you can see that this is already fitting reasonably well. And so what happened is that if we do this twice, so this was initial parameterization, and you see the huge error bars that we have on every one of the kinetic parameters, because we really didn't know many of these parameters very accurately. After 2 rounds of pulses, we have a much, much tighter sort of range of parameter values for each of the different rate constants in our model. Now, what that means is we can now predict sort of the outcome of a reaction based on multiple objectives. So we can say, I want to have a specific ratio of UTP versus GTP versus ATP, and I want an overall conversion of this compound over here of minimum, say, 70% or 60%. So we have a multi-objective function that we can then sort of run again, and we sort of see thousands and thousands of different possibilities of sort of different input combinations of reaching this particular output combinations. And you can see that we can do our selection. And then what we find out is that indeed, if we sort of have a UTP, ATP, UTP, GTP range of 3 to 1 to 1, or 1 to 1 to 2, or 1 to 2 to 3, we can predict this very accurately, while at the same time we have a certain conversion efficiency. It turns out that if we are really low concentrations, then actually our model breaks down, probably because our HPLC data are not sufficiently accurate at very low concentrations. So we do need to have a certain sort of input concentration that is above a few millimolar. But if we are able to do that, then we can now predict the output using sort of a certain input combination. So this is just to briefly show you how we are using sort of our optimal experimental design and flow chemistry to have a general platform now where we can sort of, in a way, predict the outcome of complex reaction networks. We have a quantitative analysis on the dynamic response of enzymatic reaction networks to external changes. We know how to build this maximally informative data set, and we have a high level of control of complex ERNs. And for the discussion later on, I'm really happy to discuss the next step, and especially what are the networks that really need this very advanced way of finding the dynamic response to changes in the environment. How do we optimize things? And also, what kind of challenges do we have in terms of what kind of objective functions do we need to define? Is it just yield, or is it cofactor use, or is it yield plus cofactor use plus a certain combination of a mixture of output concentrations? Those are the kind of questions that we are looking at, and I hope to discuss with you what kind of good targets would be for our method. With that, I'm happy to stop sharing and open the discussion.
Thank you very much, Will, for the presentation. Well, as just said, we're looking for questions. Maybe until the first one is typed in, I have a very simple one. Well, I'm not sure. You said that you want to parameterize the kinetic model. So you put up a kinetic model in the background. What do you see? What is the advantage of taking a kinetic model over just a completely black box, just taking any KI tool?
I think it's complementary to that. If you do a completely black box, you will never find out why it responds in a certain way. I should have mentioned we start with a certain model because we think we know what the interactions are. What you can do is, as part of this optimal experimental design iterative process, you can actually also test different types of models. So I can have multiple models, automatically generate them if you wish, where we add many, many new interactions that we think might be happening, might not be happening. And your sort of experimental input pattern that you get and your output sort of pattern that you get, you can match against many of these different models and see which model most accurately predicts the sort of input-output relationship, which doesn't always necessarily mean that this is the best model, but we then simply say, well, the most simple model is the best model in that case. So it's also a way of extracting hidden interactions that are not immediately clear to you. So that's where I think it sort of complements an AI-based approach, where in the end you don't really learn about those kind of hidden interactions.
Okay, thank you very much. That clarifies this point. Okay, I think in the interest of time, we move on. So I would ask Christoph Winkler from the University of Graz then to share his slides, and the stage is yours. I can see the slides. Perfect. Thanks, Christoph.
Well, thanks. Thanks, Wolfgang. And also, I'd like to say thank you to everyone involved in making this workshop happen. Special thanks go to Miriam. Well, as Wolfgang already mentioned, I'm going to talk about our experiences when we tried to get a light-dependent enzyme to work in flow. First, I'd like to dive a bit into photobiocatalysis. So we recently distributed the growing field of photobiocatalysis into 3 subfields. The first one would be the photoenzymes, so enzymes that contain the chromophore already in their active site. The second one would be the coupling of classic photochemistry, photocatalysis with biocatalysis. And in the third pillar, natural photosynthesis would be coupled to biocatalysis. And over the last couple of years, our working group contributed to pretty much all of these systems. Today, I'm going to talk about the enzyme that is down there. So the fatty acid photodecarboxylase. I also like to mention that, you know, in order to get photochemistry running in your lab, you need some equipment, and we also contributed to that. What you see here is a small photoreactor that allows us to run photobiocatalytic reactions under very reproducible conditions in parallel. We have been using this for most of the stuff that we have been doing in the field up to now. You see here we can run several samples at once. We also use this reactor for upscaling, and we also use it for flow chemistry. And the first part of the presentation here was done actually in a setup that's similar to this. Later on, we also got a new reactor, which is from Lumidox, an illumination plate that allows us to do photobiocatalytic reactions now in a 96-well plate format, but also, again, use it for flow chemistry. And the second part I'm going to talk about was done in this device. So, well, this is the enzyme we were interested in. It's the fatty acid photodecarboxylase. It's a light-dependent enzyme that is able to decarboxylate fatty acids to produce the corresponding alkanes. And when it was published a couple of years ago, the community immediately saw its potential as a potential application to produce biofuels, especially drop-in biofuels from renewable resources. What you see here is the substrate scope of the natural CVFAT, the natural photodecarboxylase. So it is more active for medium to longer chain fatty acids, and it also allows some substitutions like a terminal carboxylic acid or an alkane in the chain. Supplying decoy molecules is a trick to get it also converting shorter chain fatty acids, and the enzyme proved to be quite mutatable. So using the correct variant, a huge variety of different substituents can be introduced. The enzyme was shown to be have some stereoselectivity, even larger substrates are converted. So besides its potential for biotechnology, there is also potential for organic synthesis. Well, it sounds like a really ideal enzyme, but there are two, I would say from my perspective, two major issues. The first one would be the enzyme's inherent photolability. So as soon as, if there is no substrate present in the active site, well, and the enzyme is excited to its first excited state, it starts to attack itself and thereby die. And another issue is actually inherent to all photochemistry. So if you want to scale up a photochemical photocatalytic process, you need to deal with the problem of light, the limited light penetration into your reaction vessel. This is just a very easy example using the Lambert-Beer law. If you have a 100 micromolar FAD solution, this is for photochemistry quite a low concentration. For photobiocatalysis, I would say is reasonable. Well, if you have this 100 micromolar FAD solution already after 2 centimeters of penetration depth, only 0.5% of the initial light intensity is left. So you see there is a huge problem if you want to work in big reactors. We decided to tackle both problems. We hoped to tackle both problems by getting the photodecarboxylase to work in continuous flow. And well, this is what I mainly want to talk about. What is here on this table is, so actually, our starting point was we wanted to have a heterogeneous process. So we wanted to apply the photodecarboxylase as a stationary phase. And for this, we went to immobilization. We tested a whole variety of commercial supports. As you see here, we used supports that would interact with the enzyme via electrostatic or hydrophobic interaction. We tested different supports that would allow a covalent binding of our target enzyme. And we also tested quite intensely, actually, the ECG method that would bind the enzyme's histidine tag via metal affinity. It took the student involved in the project quite some time to get good conditions for immobilizing CVFAB using the ECG beats, but we believed it's worth the effort because in contrast to the other methods, it would allow binding of the target enzyme directly from the enzyme, from the lysate. So we could skip the step of purification. Well, in the end, the student succeeded and he got an even active preparation. However, he then also tested immobilization of the purified enzyme, and like this, we got the highest activity of an immobilized CVFAB. However, when he then proceeded and used this preparation as a catalyst in heterogeneous continuous flow, he quickly found some more problems. There was enzyme leaching, and there was, well, the support was not compatible with our substrate and the product. So we decided to go on with the second best preparation, which was the covalently immobilized CVFAB on this UPACHIT catalyst, UPACHIT support. Well, using this and running a couple of experiments in continuous flow, using this as a solid stationary phase, this was the outcome. So what you see here is a flow experiment with 20 minutes of residence time. And what you can see, flow is not the solution for the limited photostability of our catalyst, as the activity goes, or the conversion goes down over time. Even reducing the residence time basically gave the same result. So the catalyst slowly dies, the immobilized catalyst slowly dies. Maybe it's worth mentioning, we got the highest conversion at residence time of 20 minutes, but we got the highest overall product formation until the catalyst was destroyed itself using 10 minutes of residence time. Anyway, we then decided to go one step back and to also supply the catalyst continuously. So we used the photoreactor for the efficient illumination of a reaction mixture, but we would provide the CVFAB, so our catalyst, also together as a mixture with our substrate. And doing this, we optimized the reaction parameters. So we found the optimal photon flux for our setup would be 10 microeinstein per second. We found that 10 minutes of residence time was sufficient for our purpose. We found that we need a quite high enzyme concentration for our best conditions, which would be 16 micromolar of CVFAB in solution. And then we demonstrated that this homogeneous process was stable over the course of several hours, giving a stable 65% of conversion. And in doing so, we were able to establish the highest yet published space-time yield of a CVFAB catalyzed process of 5.7 grams per liter. And definitely there is room for improvement, especially regarding the enzyme concentration or even the enzyme preparation. This was done with purified enzymes. CVFAB is reported to be even more active if it's in the stable environment of its cell. So that's not the end of the story, but still it's a quite significant result. We then even transferred these. We then even tested many other substrates, so basically fatty acids of different chain length. And what you can see here is that the conversion in principle matches what has been reported for the batch process. So the batch process seems to be very well transferable to these new flow conditions. Well, as a next step, we decided to make the system a bit more complex, and we decided to add one step before, namely, we decided to use an immobilized lipase to also hydrolyze a triacyl glyceride. Overall, this cascade would then allow to use these natural triacyl glycerides to end up after first hydrolysis and second decarboxylation with the molecules of interest. I'm not going to show you the whole story, but the first step was to find a suitable immobilized lipase. We tested what we had in-house. We found that the amano PSD would be would show the highest activity for our purpose, but it was not compatible to flow conditions. So for the two-step flow process, we used this NZL107 IMB lipase. This is what the setup looks like. So first, we would start with a pump that pumps triolein into our reactor, and in a T-shaped connector, it would be mixed with a mix of DMSO as a co-solvent to solubilize the substrates, then later on for CVFAB and a buffer. This would be pumped through a column filled with our heterogeneous lipase. At this stage, the hydrolysis would happen. Then afterwards, again, using a T-shaped connector, CVFAB would be supplied again as a solution in buffer. At this point, the overall mixture would be diluted one by one. And then finally, this mixture passes the Lumidox lead plate in order to let CVFAB do its photodependent reaction. So what you see here are the first results, and we are already able to produce roughly 1 millimolar of product concentration. However, I really must emphasize that none of these steps has been optimized. We see activity goes down. This is probably due to deactivation of the lipase. We definitely can work on the co-solvent, so there is still some work that can be done with this process. Well, finally, I'd like to say thank you, first of all, to the EU for funding the CLASSI project, and most of the results shown here were done within this CLASSI project. Then everyone from our working group involved in the CLASSI project, especially Wolfgang, the head of the working group, Stefan Simic, who did most of the results that I presented here, and Matthias Pickle. Then our cooperation partners from Radboud University, and finally, I'd like to thank you for your attention.
Thank you very much, Christoph, for this presentation and showing the challenges with light inflow and also with the light-dependent enzymes. So as you have seen in the chat, if there are any questions, please type them in. Maybe, Christoph, I can ask a short question. Well, you showed and you mentioned, and that's why you changed then the setup, that the enzyme was rather unstable under light. Do you see a chance that this stability or could be improved, or would you see that as an intrinsic problem?
Well, I see, without going too deep into the enzyme mechanism, I see many things that can be done in order to stabilize the system. Our idea in this case was, it has been shown that that the enzyme deactivates if no substrate is present in the active site to facilitate to act as a reaction partner with the flavin in the active site. So our idea was, if we continuously provide the enzyme with more substrate, then well, this self-destruction would not happen. But you know, the excitation of a flavin cofactor actually happens at the speed of light, so that was faster than our flow rate could provide new substrate. I think the next step to do would be to investigate what actually happens in the active site if this flavin cofactor really attacks the protein, and maybe then enzyme engineering could be a solution. But there are also other options. Maybe there are options to actively add another agent into the solution that would be able to act as a quencher of the excited state of the flavin if no substrate is present. So I think there are many ideas around, and I think this is really the main issue that needs to be solved before this enzyme can be scaled.
Christoph, there is one question in the Q&A from Selin Kara. Thank you for this nice presentation. How was the longer stability of the enzyme under flow with light? I mean, you had it probably on the graph.
Yes. Thank you, Celine, for this question. So I cannot give you good numbers of a half-life time because we simply did not measure. So we had this pragmatic approach of running it and just see how well it gets. But if I can go back to the one slide where we saw the deactivation over time. No. So you see, well, depending on the reaction conditions that we used, we basically lost activity after 2 hours, but usually also from experience from batch, activity can be gone even before. But I'm aware that you found a nice trick to make the enzyme a bit more stable using ionic liquids. But I have to say, we have not tested this in our system yet.
Okay, thank you very much for the answer, and thank you for the question. And as we can see, there are options probably to address that. Please, for the next talk, I will move on in the interest of time. Please type in the question in the F&A. Forward to the talk of Inside Biotech. The talk is given by David Rura-Batrosa. Title is on the screen: Immobilized to go further. Bioinformatics role in advancing enzyme immobilization. I'm looking forward.
Thank you very much, Wolfgang. And yeah, I want to thank also the organizing committee for inviting us to talk here. I titled the presentation Bioinformatics Role in Advancing Enzyme Immobilization, as you well said. And you're going to see that's the core technology that we want to push at Insight. We believe that we need to immobilize to go further if you want to use biocatalysis at industrial scale. And we think that bioinformatics has a big role to play on making this whole process more efficient. Just because we're quite new, I just wanted to have an introduction slide where you can see where we're based. So we're a spin-off of the University of Bern. We are still based in the DCVP building in the beautiful city of Bern. And here you have the 3 co-founders. It's myself, who I'm now the CEO of the company, Dr. Honeyman, and Professor Francesca Paradisi, who is the key scientific advisor and also one of the co-founders. As you can see, we have been founded only in 2022, but during this year, we already had received some funding that allowed us to expand the team. You see, we have the team Valentina here, with Valentina Martini being our project leader, and Valentina Folli, that is the research assistant. And the main goal of Insight, as I said, is to try and de-risk enzyme immobilization and make immobilized enzymes accessible to the industry. Now, the problem that we see with enzyme immobilization is that since the 1970s, but also much before that until now, enzyme immobilization has been a technique that has been tied to a trial and error approach. I think Christoph presented it very well. When you have an enzyme that you want to immobilize, your only options is you try a different set of resins with different chemistries in there, and you hope that it has a significant immobilization yield and that it has the activity that you want. If that doesn't happen, what you have to do is just go back, try different supports, different chemistries, until, and you repeat this process until you have an enzyme, a hit, that has the immobilization yield that you want. So that means the amount of protein that you want bound onto the support, a high recovered activity typically, and the correct stability for your desired use. Now, this problem that we're facing with protein immobilization is not a new problem in biotech in general. It's a combinatorial problem. And I chose here just one example to illustrate that. Protein engineering in the early 80s or early 60s to 80s when it started was also a combinatorial problem. And what people were doing is just taking an enzyme, trying to mutate it through synthetic diversification towards different variants of those enzymes, and then screening them in auxotroph strains, where they could see if some of the variants that they have produced were better than the wild type. And they kept repeating this process back and forth until you find something that has either the activity or the selectivity or the stability that you want. Both these techniques, enzyme immobilization until now and protein engineering until, let's say, the early 2000s, were based on a trial and error approach, were costly and time consuming, and were mostly unpredictable. But we can all agree that protein engineering has gone past those limitations. From the early 2000s, there were, or I've highlighted here, three main advances that have helped massively protein engineering. The first one is high throughput screening, which allows to obtain and process the data much faster. The second one would be these tailored bioinformatic tools for the process of protein engineering that allows us to recognize, understand patterns in this data that we have obtained. And finally, if you combine both, what you can do is a data-driven development, where you can have decisions that are not random anymore, but you can take precise decisions towards your final goal, which at the end enhances your success rate, obviously. So the idea of Insight is exactly that, to untie immobilization from its current unpredictability by using these three main ideas that I have already presented. With the idea that we can avoid all the middle part, that is the tedious and time consuming and cost of the process, and we can go directly to these immobilized enzymes that we can apply for the synthesis of high value chemicals. In the interest of time today, I will just be talking a bit about the bioinformatic tools, but if anyone is interested in the other two, we can discuss further. So for the bioinformatic tools, I'm going to start talking about CapyPy, then I'm going to give you just a hint of what we have, what we do at Insight, and then ongoing development is going to be very brief, just as a take-home message and to keep you hooked into our company. So in fact, Insight started with the development of CapyPy. CapyPy is a software that is targeted to scientists that want to tackle immobilization and already have some experience of it, and want to use some bioinformatic tools to try and rationalize their process. It uses readily available data and open source tools, so it's completely free to use, and it's a protein-centric analysis. You can see here that is composed of 4 modules, and today I'm just going to be focusing on the surface and cluster module because I think it's the most interesting one, because it's the one that is trying to answer the question of what drives immobilization. From our perspective, what drives immobilization from the protein point of view, it's its surface, and that means the available chemical handles that you have on the protein. How these chemical handles are organized in the 3D structure, so the relative position that they have if they're clustered all together or they're very sparse, and also the distance of these clusters to these structural motifs or important motifs that the protein has, such as the active site or the interface. I'm just going to show you now a quick example of how that looks with a real protein, and then I have 2 concrete examples that you can see. They're very simple, but you can see very clearly what we mean by how can this affect the immobilization. So in reality, now, this is how it looks when we look at the surface of the enzyme. As you can see, just looking at the surface is quite messy. You have plenty of different functional groups, plenty of different amino acids that are exposed. But it's more interesting when you graph it because you can compare protein by protein. So for example, this is interesting to see if your protein will have any lysine exposed, that is the residue that is most commonly used for immobilization, also how negatively charged your surface is. So we use that in the first step. Then what we do is the cluster analysis, where you can see now that that becomes much more interpretable for our human mind. You just have negative clusters in red, and for example, the positive clusters in blue, and you can see that now we have much less, so we can understand much better what's happening. And then the third part is how close those clusters are to interesting functional sites. So in that case, for example, with this protein, you have this negatively charged cluster that is actually quite close to the entrance to the active site. So if we were to immobilize through that cluster, probably we would have an inactivation of the protein because we're not letting the substrate in. Just an example now on the importance of this cluster location. This is a protein that you might be familiar with. It's the formate dehydrogenase from Candida boydini. And this enzyme, when you try to immobilize it through epoxide handles, and therefore using the amino groups that you have on the surface, especially lysine amino groups, you have very low recovered activity. Now, when we look at the position of these clusters, you can see that both clusters are at each end of the monomer. So it's not crazy to think that when you're immobilizing that protein through those regions, you're straining the tertiary structure and therefore immobilizing just the monomers and an inactive version of the protein. Now, when we immobilize that same protein with the presence of some amino groups, either through an amino epoxide or through a polyamine, such as the polyethylenimine, you have much better recovered activity. And you can see here that the negative clusters of that protein are indeed in the interface between the two monomers. So what can happen is that you kind of are threading this protein together and maintaining its quaternary structure, and therefore maintaining much better its activity once immobilized. One of the arguments that we've had for this approach is that we're looking at static structures, and that's completely right. We're looking at the static structure, just the PDB file of the protein. But now we are not limited anymore to just the snapshots. We can go to the full movie. We have now developed short molecular dynamic simulations that can be run in any laptop, and that allow us to also look at the dynamic exposure of the different residues throughout, in this case is 5 nanoseconds, but throughout the process of immobilization, and understand if the protein is actually changing from its crystal structure to its more mobile structure. An example of that is lysine dehydrogenase that we're working with. As you can see here, it had a better recovered activity with agarose, which is a more hydrophilic polymer. Judging by the structure and also the surface of the enzyme, it's not strange that it has high recovered activity, as almost, well, a bit more than 40% of the surface is occupied by hydrophobic residue. So you can understand that when immobilized in methacrylate, probably it just suffers from denaturation kind of process. But it's more interesting to see that when we look at the dynamic exposure, we see this tendency is even aggravated. We see that, for example, aspartic acid, which is a charged amino acid, just decreases in exposure, while there are several hydrophobic residues that are increasing in exposure in here. So as a conclusion, and just more than happy to answer questions if there was any, at Insight, we use these data-driven approaches to tackle enzyme immobilization. We're not limited only to the analysis that I've showed here, but analyzing the protein really helps to consider the compatibility with the material and also the effect of the binding, the effect of the material in binding the protein. And what we have down the pipe, and I hope that's exciting for all of you, is we're developing full workflow that we call TIMOFinder that will finally give you an immobilization probability directly on your protein. That's going to be for academics and companies. If they're interested, we can talk later. And also with the data that we have produced, we're almost finishing the validation of a machine learning classifier that allows us to, just from the protein sequence, understand if the protein will behave well or not with a certain immobilization chemistry. So with that, I just finished the presentation. Happy to take any questions. And if anyone has any other questions apart from the time that we have here, feel free to reach out to us. Thank you.
Thank you very much for your presentation. Please use the question tool. I have one here anyway in the chat question. Great to see the capabilities of the software. How would you direct the chemistry to a specific lysine, for example, if there are many other sites?
That's a really good question. So one way that we have is you try and understand where that lysine is positioned. And for example, if you have a high density of negatively charged residues in there, then maybe a support that can interact firstly with those will kind of direct the protein. It's true that without touching the enzymatic surface, you can just direct it as much. So it's not fully customizable yet, but then protein engineering could also be a solution there to try and direct it even better.
Okay, thank you for the answer. There's another question from Matthias Bickel. Thanks for the very nice presentation. Does it also consider the oligomeric state of the protein?
Yeah. Thank you, Matthias. So yes, the software does consider the oligomeric state. You can either just import your own PDB file or create a model. And obviously in the model, then you're limited to whatever closer homolog we can find. That's the oligomeric state that we're gonna assume for that protein.
Thank you very much. Well, I have also a question. I mean, I like very much your approach to say that we need to address the different clusters on the protein immobilization to simply have it better immobilized. On the other hand, you showed a few slides later these molecular dynamics, and so stating that the enzyme is not static but is highly flexible. There's always the thought that when you immobilize, you limit flexibility, you limit movements, which might be important for the catalysis. Do you have any experience when you do this now 2-point attachment via different groups, how that will affect the activity?
We're trying to look into how exactly we're affecting the flexibility of the protein once immobilized. You're completely right that affects catalysis, and that's a trade-off that we always have with immobilized enzyme. You're going to have something that is stable and reusable, but probably is going to be less active. So it's always to find the sweet spot between the two. I hope that soon we can have an answer to exactly how much the flexibility of the protein is affected. But the flexibility that we're analyzing here is more to understand, are the clusters really there all the time, or are the transient clusters, which amino acid is exposed and for how long? So we're trying to look at this from a more dynamic point of view rather than just from the PDB file. But yes.
Thank you very much. I'm very much looking forward to test the tools. So thanks for your presentation. And we move forward to ancient biocatalysis, and Mattia Lazzarotto and Stefan Bayer will give the presentation on strategies for optimizing biocatalytic cascades, current challenges, and future perspectives. So the stage is yours.
Yeah, thanks a lot, Wolfgang, and thanks a lot for having us here today. My name is Stefan Bayer, and I'm the CEO of Ancient Biocatalysis, which is a spin-off of the University of Graz. And here at Ancient, we are convinced that actually exploiting the natural compatibility of enzymes in biocatalytic one-dot cascades is one of the most proactive and beneficial ways of using biocatalysis today, because it offers really enormous potential for saving costs and resources. But we all know that there are really a lot of technical challenges to be solved or to be mastered on a way to successful industrial deployment of such cascades. Talking about enzyme immobilization and stability problems, some of which can actually be overcome by flow processes or immobilization. But here in Ancient, our goal is to establish a set of tools and approaches for the efficient development and optimization of mostly batch cascade processes that help unlock their benefits for manufacturing value-added chemicals. So therefore, it is now my pleasure to hand over to our CSO, Mattia Lazzarotto, who will tell you more about the cascade case studies and optimization approaches that we have been looking into in ENCIAN in the last month. So, Mattia, please take it away.
So hi everyone, thanks again Stefan, and thanks also Wolfgang for the kind introduction. So in the next 10 minutes I will guide you through some strategies for optimizing biocatalytic cascades and some approaches that we have taken here in ENCIAN. So actually the problem was already introduced today by the first speaker, but just to mark it again. So here an example of a biocatalytic cascade, a very simple one. There are only 3 enzymes involved, but when you consider also that there is cofactors, co-substrate, substrate, and other salts, together with the pH and temperature, this gives a lot of parameters that have to be tuned in order to basically optimize the condition in which the biocatalytic cascade run, and we want to optimize all these different parameters in order then to find which are the best conditions that can maximize the performance of a given biocatalytic cascade. So with all these reaction parameters, we end up with a multidimensional design space in which there are probably a lot of local minimums, some local optimums, but also what we are interested in to find is this global optimum, which are the reaction conditions which basically maximize a given key performance indicators of our choice. But the problem is that already with the simple examples, there are already more than millions of possible configurations in which this cascade can be run. And the scientist in its laboratory basically can only assess a tiny fractions of all these possibilities, but still would like to identify what are the best conditions that are needed to run the cascade then in an industrial fashion. So in order to optimize this, in order to optimize biocatalytic cascade and more in general reactions, there have been already different approaches. And in this slide, I will just go through a couple. So let's say the historical one, the most old one, is basically the optimization of one factor at a time, where it's basically we use just literature and intuition, and thanks to a trial and error approach, we just go and optimize one factor at a time. Basically, I don't know, first we fix the pH, then we fix the temperature, then we fix the given concentration of one enzyme, and so on. But by doing that, we miss out all the basically interactions that all these parameters have in actually in the batch when we put everything together. Another approach is the use of design of experiments, and software has been developed in order to make it easy for scientists to use this technology. And basically, we can here run like an initial sampling of our design space, and then use statistical methodologies to basically have a surface, have a response surface, and basically use this to basically try to predict experiments which can maximize the performance of our cascade. Then we have another approach with kinetic modeling. We have heard already today, and this gives basically a physical understanding of our system, where we basically want to model each step, each enzymatic step with the Michaelis-Menten equation, and build an overall kinetic model of our cascade, and then try to understand what are the conditions that can maximize, I don't know, the yield or any given key performance indicator which we are interested in. Further, with the advent also of machine learning technologies, there are also data-driven optimization, which take advantage of high throughput experimentation, and also with the automatization and miniaturization in order to run many experiments at the same time, and then find actually the one which perform best. Last but not least, also self-optimization strategies, in which basically we try to do an optimization over cycles, and at each cycle we try to improve the conditions in which the biotransformation, this biocatalytic cascade is running, and we complement this with the machine learning in order to basically predict at each cycle the experiments that we want to run. So at ENCIAN, we were interested, especially these last two approaches, because we believe that having a tool which could be agnostic to every biocatalytic cascade and could be fast and reliable for the optimization of such system could be quite interesting and could solve the problem for this optimization. And there are already a couple of examples in literature when mainly these approaches were used in other fields, for instance, in chemistry and flow chemistry by the group of Professor Bohr, also in synthetic biology by the group of Professor Follon, and also in material discovery. And initially, when we approached this problem, we actually started from this workflow that was developed by Professor Follon, which was basically an active learning workflow, which is basically a design, build, test, learn cycle in which an initial data set of reaction is decided to be run, can be random, can be also a different data set. And thanks to liquid handling, basically liquid handling robot, we can perform this reaction, and after they are run, we can analyze them with the high throughput techniques and use then a machine learning model, train a machine learning model with the results, with the data that we have obtained, and use this machine learning model to predict new reactions can improve a given key performance indicator of our choice, and do this over cycles till we reach a satisfactory level of optimization. So here I will just give you an example how we use this for a single objective optimization. So we took as a model cascade the one that I have shown before. It's only important to know that there were 6 continuous variables and 2 categorical variables with a design space given by these ranges. So here you see the graph that of the result of the optimization that was performed after 5 rounds of active learning. We started basically with a random sampling of 400 initial data points, and over 5 rounds we were able to obtain a satisfactory level of optimization with this biocatalytic cascade, optimizing towards this key performance indicator that we chose as a yield square divided by the enzyme loading. Moreover, it's also interesting to see that in this case, we were not able to not only to perform an optimization, but we also used the active learning in order to select a given enzyme, one tyrosine ammonia ligase, which is this enzyme that we see here in the middle of the cascade. And by providing 3 different alternatives with the machine learning, we were able to predict which of these enzymes could fit best in the cascade and provide the best results. Okay, so basically I've shown you now here a proof of concept for an optimization on a single objective, but in most cases in biocatalysis or in chemistry, we are always interested in having multi-objectives. Basically, we could decide to optimize towards a high yield, but also reduce the enzyme loading, also optimize towards chemoselectivity and anti-selectivity and so on. So it would be interesting to have a tool with which we could use to optimize towards multi-objective. In this case, the problem is a bit more complicated because we won't have not a single solution, but we will have multiple solutions. Basically, you have probably heard of this set of multiple solutions, which can be called the Pareto front, which basically determines which are the best solution of our system. And we can basically describe solution as dominated if there is another point, which is basically better in all the other objectives. So by using these, we can sort all our data in solutions which are dominated or non-dominated. So for tackling these, we basically took advantage of this Python epsilon Pareto active learning algorithm that was developed a couple of years ago. It was only used once for a material discovery approach. In this case, it's a very simple active learning like a workflow, but at each iteration, we are not interested in basically suggesting new reactions that could improve the performance of our key performance indicators, but we just want to compute a very accurate, more and more accurate Pareto front to be able to basically classify all points, whether our Pareto or basically as discarded and not interested, basically. So for a first basically case study for this multi-objective optimization, we decided not to use any wet lab data, but just carry out a synthetic experiment, an in silico experiment with synthetic data, which were obtained from a kinetic model that was generated already by the group of Armour in the cascade that you see here, which is a 7-enzyme cascade. So we took basically this kinetic model just as an experiment, and we could basically use this as data. And to simplify it, we kept the variable only the enzyme concentrations, which gave already almost 100,000 possible combinations. And we did the first optimization with 500 initial data points, and at each iteration, the algorithm was basically sampling 20 new points, and we tried to optimize for maximum yield and to minimize the enzyme loading. And here I just show you our first experiment that we did. Basically, in these graphs, you see here the orange points, which are discarded and not interesting, and the blue points, which are the points that the algorithm sampled and wanted to know the actual value. You can see them as experiments, and the green points, which are points which identify as Pareto optimum. And after 58 iterations, the algorithm identified more than 5,200 Pareto points. So here we are with a lot of points actually. So, and we could actually realize that in the right side here of the graph here, we could realize that there is a high error at this point compared to the actual kinetic model. But these points that lies up here were actually quite in good accordance with actually what the kinetic model was indeed predicting. So there is still a lot to improve in this, but this is how we want to approach this problem and try to implement this for the optimization of biocatalytic cascade. So all these optimization workflows that I've showed you, they are embedded a bit more bigger frame, which is the development of a self-driving lab, which combines automated experimentation like enzyme expression, cascade assembly, and running actual product transformation with machine learning in order to speed up the prototyping of ideas here at Enzian. So with this, I already come to the acknowledgement. Yeah, first of all, I want to thank the group of Professor Wolfgang and this group, because most of this work was carried out in his laboratories, all the supporters of Enzian in the past 2 years. And then, yeah, all my colleagues at Enzian and you for your attention, and also the organizing for inviting us today for this for this workshop. And please feel free to ask any question and also reach out to us if you want.
Thank you very much, Mattia, for your presentation. Thanks to Stefan also. There's a question, great talk and impressive results. For the active learning, you use the enzymes as a variable. Are there ways of automatically coupling this to protein database so the algorithm can search for more suitable enzymes?
Yeah, I am sure there are ways that this could be done. Just because in this case we did not include, let's say, the sequence of the enzyme as you know in the workflow. It was just basically okay, enzyme A, B, or C, and then the algorithm could basically just either use one or the other one in each cascade. So. But of course, it will be, let's say, this is a future vision. This would be great if we could have a workflow that could already look for new potential enzyme and whether score them with a given score and check whether this could fit in the cascade or not. Yeah, okay, this would be really cool and really potent, and we hope that in the future this will be possible. But yeah, I'm sure that there are ways to do this.
Yeah,
thanks.
it would definitely significantly speed up as well and would have a significant impact. Right, I don't see any further question at the moment, and also that fits to the time schedule. So thank you very much, Matthias. I suggest that we go forward to the next presentation by Javier Ibanez from Enzyme. The modeling and experimental strategy for the development of scalable enzymatic reactions. It seems that sharing is working. Yes, perfect. I see the slides. The stage is yours.
Excellent. Thank you, Wolfgang, for the kind introduction. Thank you also, Marian, for the organization. I'm very happy to be here and talk on the name of enzymes. So I am Javier Ibanez. The title, as you well said, is about, I'm going to talk about modeling experimental strategy for the development of a scalable enzymatic cascades, and I will give a bit more detail what I mean by a scalable. So first, a little bit of introduction of enzymes. So enzymes is a startup company, it's a biotechnology company founded in 2014 in Sweden. We are based in Stockholm. As of today, we are a little bit over 50 total staff members, and most of us work in R&D. So it's like 36 out of those 50 employees are R&D workforce. At the origin, the company was founded around ECG, a technology to immobilize enzymes. One of the materials that previously Dr. Christoph Winkler had tested on one of his experiments. But as of today, the company focuses on a more comprehensive solution. So we do not only develop catalysts, but we develop full chemical processes. So this is a little bit how the company is structured, the R&D function of the company is structured, and a little bit how the timeline of our projects work. So we start with an enzyme engineering team that would scout for enzymes that are active for certain target reaction, that are stable at certain reaction conditions. And this enzyme, once identified using high throughput tools, it is transferred to a biocatalysis team that will optimize the immobilization of this enzyme in our carriers. Then it gets to the process engineering team, which I'm a part of, and I will be focusing on this bit of the process, on the reactor engineering. And based on our findings, we provide feedback to help optimize the catalyst, and we also feed forward information to the downstream engineering team that will develop all the downstream separation. And they are very dependent on what is coming out of the reactor, of course. So they depend on our results to develop an efficient process. Finally, when we are happy with the solution, we proceed with piloting and scale up of this process. So at this stage, we need to be sure that we have done a good work all on the way. So I will focus on this bit in the reaction engineering and try to explain which tools we use to provide information backwards to improve the catalyst and forward to downstream engineering teams. So a little bit the workflow. I won't go so much in the tools. I'm happy that in the previous presentation, there's been a lot of magnificent tools on kinetic modeling by Wilhelm, and I will not go into the details of it, but just how we use them and how are useful for us in our process. So a little bit the workflow at the reaction engineering stage. We receive a catalyst that works, and we don't know a lot about how this the kinetics of this catalyst, etc. So before starting experimental work, we take prior knowledge that we have, and we work on a catalyst cost model that we have, and we already give us an idea of what is an operative window, which target should we focus on productivity of this catalyst to see if it's going to be a viable process or not, if it's something we want to take forward. Next step would be then to developing all the experimental work and the modeling work that will aim to understand the kinetics and the deactivation of that catalyst, to see what to have a much more refined idea of the catalyst cost contribution in industrial production. This information obtained here will be used to optimize the reaction conditions and also to give feedback to biocatalysis team to tell them the catalyst you've provided is good enough for scale up, or we need to refine that formulation. And finally, once we have all this information covered, we will update our mass and energy balances, and this will help downstream process engineering team, and we will update the techno-economic model with much certain figures on the cost of our production. So I will move to the first stage, so it's the early on, only prior knowledge situation. And I apologize, I cannot give so much detail, so I've erased the values here, but it should be enough to give you an idea on what kind of information we obtain from this catalyst cost model. So basically, we look at based on the current knowledge, which process conditions we should target in terms of conversion, product recovery on the downstream, and then we see how that puts us in terms of catalyst cost contribution in our process. So at this stage, we are a bit uncertain of what conversion are we going to be able to achieve, what recovery are we going to be able to achieve in the downstream. So we provide a window of operation that, based on the economics of that process, is viable. And this gives us an original, a first target productivity for the catalyst and a first target lifetime for the catalyst. And this we will use going forward and then validate moving into the kinetic model and the deactivation model. So once we have the catalyst, now we're able to test for the kinetics, and this is done in a very small scale. We can do this in this small 2 ml EPIs. We can test many conditions at the same time. And for this particular case that I'm presenting, this is just a 2 enzyme cascade, but already it starts being a complex kinetics. So we need to be smart and use smart tools in order to be able to identify all these parameters. So at this stage, we use a very simple, well-mixed batch reactor like this. We load our catalyst, test a number of the conditions, we estimate these parameters, and then based on whether the parameters are well identified or not, we proceed to do a new set of experiments with different conditions that will help identify the uncertain parameters. Or once we are happy with the predictionability of this model, we can use it to provide information on how the reaction conditions affect the product distribution. So as I said, now that we are happy with the model, we can, for instance, in this particular case, we observed that one of the key parameters affecting the productivity of the catalyst was the ratio of the reactants. And based on the previous information on our catalyst cost model, we had set a value for a minimum productivity that we should achieve for this catalyst to be viable at industrial scale. So this point signals this minimum productivity. And in terms for having a profitable process, we should operate under conditions that are above this line. And this will be useful now to test and to test in combination with the stability of the catalyst to choose what are the optimal reaction conditions for this process. And not only optimals in terms of results, but in terms of techno-economics of the process. So now I will move a bit at the deactivation model, and this for this same reaction. If I may go back a bit, here I just gave these dummy names to the reactants, but if there is one that you need to remember, is just P stands for product, is the product of our reaction, and UP is an undesired side product that is produced under reactant. And A and B are the reactants of this network. So if we move to the deactivation, we study this in flow. Now we need to move away from the batch reactors, and we test this in these fixed bed reactors. We use typically this configuration of several fixed bed reactors in series. In this case, it's a two enzyme cascade that is packed through these 3 reactors, and we sample before and after each of the reactors. And with this experimental setup, what we aim for is that we can see the profile of enzyme deactivation. We can distinguish between deactivations that happen through the whole bed at the same time, as it would be a thermal deactivation, that it's not triggered by any particular compound of the reaction, or a leaching, etc., something that happens through the bed at the same rate, or we can differentiate the activation mechanisms that happens at different rates at different points in the bed, and this will help to identify these deactivation parameters. So in this case, we saw there was a deactivation, and we proposed 3 different models. The first model, it's a very traditional first order decay. So this assumes that the rate of deactivation is equal across the bed of the reactor, and it is proportional to the amount of active enzyme. Second model is influenced by one of the reactants. So there is one of the reactants that we suspect might cause deactivation. So this would be the rate of deactivation. And third, we suspect that it might not be one of the reactors, but might be some side product or something that is present in the feed mixture, and that leads to very fast irreversible deactivation. So now that we have this hypothesis, we build the models using reusable code that we have built over the time at Enzyme, and we try to fit the experimental observations. In this case, the points that you observe in each of the graphs, they are the same on the 3. It's the experimental observations. On blue, we see the outlet of the first reactor, on orange, the outlet of the second reactor, and on green, the outlet of the third reactor. And we see that for the 3 models that we have proposed, we can only get a good fit for this this model in the middle, which is a deactivation influenced by one of the reactants of this reaction. And we can be quite certain that this is working, since we see that it fits quite well the 3 points in the bed, which are seeing and evolving over time. They are experiencing a different concentration of this reaction. So it is this parameter can be pretty well identified in this case. So now we have a kinetic model that tells us how the concentration of the reactants influence the productivity, and we have a deactivation model that will tell us for how long can we keep this reaction running with this catalyst before we need to stop and change new catalyst, which will in an industrial setup tell us how much is the price of the catalyst, how many kilos of product we can produce per kilo of catalyst. So what we've done here is setting up a limit where we know the catalyst has deactivated too much and we need to change it. So it's going from 2 weeks, 3 weeks, and 4 weeks. If we look previously at this condition, we see that for the third reactor, I cannot show here, but after 200, 300 hours, we are already seeing one of the reactants in excess coming out. So we have that limit of a conversion going under 90% of one of the reactants, and we decide to test what are the viable conditions to keep the catalyst activity during 4 weeks. So this is only one of the parameters we study. We identify this viable region. So we were interested in operating these reactors over 4 weeks, and we chose a condition between this yellow region. So now we are extrapolating.
Javier, please get slowly to an end.
Okay.
Your time is running out.
Apologies. So we are extrapolating here, but since we are using a mechanistic model, this allows us to test. This is validated in the experiment, and we are able to run for 1,000 hours, which is over 3 times what was previously achieved. And with this, I end. Basically, I've presented the workflow where we start with prior knowledge to develop some targets. We develop a kinetic and a deactivation model that allows to refine these targets, and we iterate on this cycle to optimize the production of the targets of the catalyst. And with this, and apologies for the extra time.
Thank you very much. Thank you very much. I highly enjoyed your talk. Anyway, as I said, you took the time of the questions. So in case of questions, please either keep them for later that we might address them in the panel discussion or contact Javier Ibanez directly. I would suggest, as we have on the program, that we make a very short break, 5 minutes, just a bio break, in case you need some coffee in between to get for the second round. Feel free to do so. I would suggest that we start again at 14:40 and continue with the next presentation. So let's continue. I'm very happy that Rob Skubert accepted the invitation and to present 100, at least on the title in the program, 100 years of immobilized enzyme. So the stage is yours.
Okay, thank you, Wolfgang, for the introduction. Can you hear me well?
Yes, perfectly.
Great. So welcome back, everyone. Yes, so today I want to take you on a small journey through time and talk about enzyme immobilization. So I'm Rob Schouwaert. I'm the managing director of Carovision, and just a quick introduction of the company. Carovision was founded some 17 years ago, and we are located in the Netherlands, in Den Hoorn, close to Den Haag, Rotterdam, and the TU Delft University. So the focus of the company is on enzyme immobilization and on enzyme carriers, which we call immobeads. So Carovision does not produce any enzymes, but we use enzymes every day, and we use them to design industrial processes. And important technology of that is enzyme immobilization. So we have various products readily available, immobilized enzymes like lipases. We have enzyme carriers, all sorts that are readily immobilized, and different enzymes. And then also there are some compounds that we make with these enzymes. But yeah, back to immobilization. So we know that enzyme immobilization has been around for a while. Well, actually, it was over a century ago, in 1916, that first scientists discovered that an enzyme could be immobilized. So these were Nelson and Griffin, and they were actually trying to purify invertase, an important enzyme that converts sucrose into fructose and glucose. And so they were investigating this, trying to purify it, and then they noticed that the enzyme adsorbed to one of the additives, the charcoal or the aluminium hydroxide, and they could actually release the enzyme back from it, and then it was still active. So quite an important discovery at that time. So if we just take the decades one by one, then that era following up that first discovery was mainly the immunologists that were active in, say, immobilization of enzymes, but then it was basically isolation of proteins. Besides the named carriers, they also use glass beads, which are a bit charged and can also bind proteins. And after the Second World War, yeah, things accelerated a little bit. A lot of interesting enzymes were isolated and also immobilized. Enzymes like amylase, chymotrypsin, lipase, first lipase immobilization, and even enzymes like ribonuclease. And the carriers that we use for these enzymes also became a bit more sophisticated. So a few, say, standard materials were added to it, like clay, bentonite, still used today, cellulose. We'll talk more about that actually. And the first synthetic enzyme carriers also saw the light of day, like the amberlite and then the famous Dowex resins. But also, not importantly, salt gel enzyme entrapment was discovered, although it took, I guess, another 30 or 40 years before there was a renewed interest in this technique, but already discovered in the 50s. Now, in the 60s, also whole cells were immobilized for the first time, which was also quite useful. Invertase, well, this was the discovery of the enzyme immobilization, but it was taken more towards industrial application. So different forms of immobilization were developed. Other important enzymes like trypsin, urease, subtilisin, amino acylase, very important. And yeah, one of the biggest products to date, penicillin G acylase for the production of antibiotics, and beta-galactosidase for making lactose-free milk. Now, the technologies how to immobilize these enzymes that were developed in that decade were, for example, the CLEC, the cross-linked enzyme crystal, so basically used for structure analysis, but the enzyme was still active, so you can use it as a form of immobilization. Polyphenol alcohol, polyacrylamide gels were also discovered to be useful for that, and a few others, among with the anion produced by Mitsubishi. Well, then the next decade, you already saw in one of the previous presentations that in the 70s there were a lot of important developments made for enzyme immobilization, although it was trial and error still at that time. So most important enzymes were scaled up, like amylase. These are all food enzymes, and the amounts of immobilized enzymes produced here was really used to make thousands and thousands of tons of products. So these were really bulk industrial products that saw the light of day. More interesting techniques were developed to immobilize enzymes, like covalent linkage that could be broken up again without destroying the enzyme. Affinity, so the histag, very important for purification, but nowadays also for multi-enzyme immobilization systems. Yeah, and dozens of other techniques, actually. Too much to mention all in detail. Now, in the 80s, well, this was the era that biocatalysis, the word biocatalysis was really, say, promoted and discovered by a bigger audience. Pharmaceutical companies, they discovered that they could get rid of a lot of protective group chemistry by using some enzymatic steps. Other advantages were mild conditions, less energy usage. And also in that decade, a very important article was published by Klibanov, who used enzymes in organic solvents. And obviously, with an immobilized formulation, you could also do this. And so this was really a turning point in history, I think. And following up that decade, in the 90s, biotechnology took overhand. And because of the availability of many biochemical techniques, one could actually change the enzyme to such an extent that one could turn it into an industrial relevant enzyme and not just depending on a suitable wild type. So that the rational design really took off. And again, the CLEC was there, but then commercialized by Altus Biologics, an American company. The CLIA was invented, the cross-linked enzyme aggregate, which meant you didn't need to crystallize the enzymes. And so these techniques were carrier-free. And actually, I also spent some years in a company called Clear Technologies, founded at the TU Delft, and back in the early 2000s. The CLIA was actually discovered by Dr. Lin-Chu Chow, who also wrote a fantastic book about enzyme immobilization, who actually inspired me to found my current company. And I took some of these overviews from this book. So if you like to deepen the subject furthermore, then I would suggest to read this book. Now, since the 2000s, we consider these the modern times, so to say, and directed evolution really provides you any enzyme that you can possibly need. Multi-enzyme systems in one pot, and that really show that biocatalysis has become matured but this is really depending on immobilization as well. Extremely porous carriers that reduce the fusion limitation to 0, but also renewable and bio-based carriers are important in these days, as well as biodegradable beads whenever they come into the environment. Now, this is a historic overview, but if you sum everything up, then if you check the annual amount of articles citing enzyme immobilization, then you can see a bit back what I just told you, that after in the 90s, biotechnology boosted, the amount of applications really took off, and you see that you really get a big increase in applications. Now, that being said, one quick overview of how do you immobilize an enzyme. So quite important, you use them in batch or continuous. For those applications, you need to immobilize it if you want to recycle the enzyme. You have all kinds of conditions, and the enzyme obviously is influenced by the immobilization, which was also nicely demonstrated in one of the previous presentations. But basically you have a lot of different functional groups on the outer surface. You can make a hydrophobic or a hydrophilic surface and combine that with different binding types like epoxides or charged groups. Affinity binding is also very important. And there you also have a lot of artistic freedom to design a new bead. Now, how do you make a bead? It's actually quite simple. You have an organic phase which can polymerize, but you stir this so that you get droplets. These droplets, they polymerize, and after that you can isolate them. Now, this looks like a powder, but actually if you put it under the microscope and zoom in, this is 100 times magnified. So these are beads on a sieve. It's easy to isolate them. And if you switch from light to SEM, then you can see more details. So this is a cross-section of one of those beads on the previous slides. You can actually already see that the pores are opening up. And if you zoom in further, you can see there are a lot of channels here, a lot of space. And actually this dot here, that is an enzyme to scale. So you can imagine how much space there is in such a microporous bead for the enzyme to move in, to be immobilized, but also for substrate to move in and out without diffusion limitation. And you have to keep in mind that at the molecular scale, the enzyme is a polymer and the polymer is a polymer, the resin is a polymer, and that interaction is quite complex. So it's really nice to see that there are some efforts being done to model this, and what exact interaction is. Although it's still a bit trial and error, I think rationalization is already to such a level these days that it can be quite well designed already, and most importantly improved. Now, just one example to wrap up. Even though 70 years ago immobilized lipases were invented, you can still develop nice applications for it, like we did for Alcresta therapeutics. We have immobilized a lipase, which is in a cartridge. It is used by people who suffer from pancreatic problems. They eat fats in the nutrition formula, but they can't digest the fats. So it is hydrolyzed by the enzyme, and these free fatty acids are absorbed by the patients, and they stay healthy. And so just an immobilized lipase, but a very interesting application. So with that, I want to close with one more graph. The market that we are moving in, based on some available reports, the current market for immobilized enzyme is considered to be around $350 to $500 million, which will double every 10 years. I would say that is my prediction based on the publications that you see here and the technology that is developed. It can actually triple every decade, which means that we are in a great expanding market, which is really in need of new technologies. So with that, I am coming to an end. Thank you for your attention, and feel free to reach out to me if you want to talk about immobilized enzymes.
Thank you very much for your presentation and this overview. I just have one very quick question for you. Well, you showed the different options to immobilize an enzyme. And when you check out, do you see, when you look at literature, is there any method which is working mostly, or is this even distributed?
Well, I think in general, you can immobilize almost any enzyme via an epoxide, as long as there are not too many, and the enzyme can retain its conformational freedom. And so as soon as you take away that conformational freedom, then you will inactivate the enzyme. So, but for a single point attachment, yeah, there is little harm done. But obviously there's also the surface area. And so there should be little interaction with that surface area. The enzyme should not absorb strongly to it, because that will also induce conformational changes.
Thank you very much. Perfect. Thank you very much for the presentation. And I'm happy and looking forward to the presentation by David Liese from Enzymicals. This last presentation has the title in the program Biocatalytic Omega-3 Enrichment in Fish Oil from Lab Batch to Industrial Scale in Flow in 3 years. David, the stage is yours.
Thank you very much, Wolfgang. I'm David Liese, so I'm the CEO of Enzymicals. We are located in the northeastern part of Germany, directly on the Baltic coast. We are a small biocatalysis group company, 20 employees, and we are involved in an EU-funded project which is called MacBeth, so membranes and catalysts beyond economic and technical hurdles. And I will take you on a little journey where all that you heard today about lipases, enzymes, enzymes in flow, immobilized enzymes, where it all comes together in a very small project that we are involved in. And I want to tell you our story. We have been in this project since 3 years. So it's as I said, an EU-funded project. The project budget amounts to 20.7 million euros. So it's a very huge project. And there are a lot of partners all over Europe involved in this project. And the project was funded or was based on 3 main cases, like hydrogen production, hydroformylation, and propane dehydrogenation. Those 3 cases already had predecessor projects where they developed a pilot plant, like you can see here, on a technical readiness level of 5. So they already exceeded the lab stage, and they formed a new consortium, and they wanted to have a little special in this project as well. And this is where we came in, and we are the 4th case, so to say. So we are the exotic edition, where we not only introduce biocatalysis into this huge consortium, but we also are the only case where we have not a gas phase reaction happening in a flow process, but a liquid phase. And with enzymes, this makes our case very special. And we also started from scratch. So the other ones already had existing pilot plants at a high technical readiness level, and we almost had to start from scratch. And this is our group. So we are the case leader of this side project within MacBeth. Also, Rob is part of our project team, Chiral Vision. The Radboud Institute in Belgium, they are responsible for membrane separation. So we are trying, or we are developing a flow reactor, a membrane flow reactor. And with that, Radboud is the expert for the membrane parts. I will not include this into this talk, just briefly mention it. Then we have Micronit from Austria, who are the engineering team in our project. They were responsible for engineering the pilot plant and building it. And then we also have Polytechnico, who will do some modeling activities, and Solutex, which might be a possible end user of the technology that I will present now. So the main idea that we have in our BOC case when we started the project is to utilize both enzymes and membrane technology to process fish oil in such a way that we have an omega-3 concentrate at the end of the whole process. And how we do it, I will show you in the next slides. So first of all, omega-3 fatty acids, there are some omega-3 fatty acids already containing in the fish oil, and fish oil has already like 30% omega-3 fatty acids. And the main fatty acids of interest are icosapentaenoic acid EPA and also DHA. So they both play a very crucial role for health benefits like the cardiovascular system, the eyesight, or the function of the brain. And the problem with those two fatty acids is that in our recent nutrition, we do not consume enough of those fatty acids. In contrary, we consume omega-6 fatty acids in such amounts which is not beneficial for us, and we should be consuming more of those two fatty acids because they enhance the health in our body. But the human body cannot produce them by itself, so we have to either eat fish or take food supplements like omega-3 capsules or algae equivalents. So as you all know, fats and oils, they remain of triglycerides. I highlighted here some of the triglycerides. EPA and DHA are highlighted in yellow. Those are the longer chain fatty acids compared to the other ones that are also contained in the fish oil. And if you add a little bit amount of ethanol and the right enzyme, then you get some transesterification, where you build up a lot of ethyl fatty esters, which are detached now from the glycerol backbone. And what you also have remain are diglycerides, or for example, here's some monoglycerides and also still some triglycerides. And the glyceride fraction is now enriched in omega-3 fatty acids due to the selectivity of the enzyme. And in our project, we screened a vast majority of all the lipases that are available. And here you can see, for example, for lipase A, that EPA and DHA, they are enriched in a very nice manner, while the unwanted fatty acids are decreased in concentration. And with this, we were able to showcase the selectivity of not only this lipase, but another one, which is also enriching DHA and EPA, but not so good to unsaturated fatty acids. And another lipase, which is not so very selective to EPA, but more so to DHA. So it is very crucial to look at the enzyme and its selectivity and choose the right enzyme for your application. So once we performed the catalytic reaction, we do not aim for full conversion, otherwise all the fatty acids will be present as ethyl esters, and we do not have an enrichment step caused by the enzyme. Instead, we have this very rather complex mixture, and now the membrane comes in place, and the membrane is able to selectively sort out the fatty esters. And what is remaining is then the glyceride fraction, where, as I said, from the fish oil, which already contains 30% of omega-3, we are able to increase the amount of omega-3 fatty acid in the remaining glyceride fraction to 50% and even higher. So in our project, we started from very small batch experiments, as you can see on the left-hand side. We then found out the kind of optimized reaction conditions, and we were able to scale it up first to 1 liter and then 6 liter in a steered tank reactor. And we were very happy to see that when we applied some first flow experiments, that later on, also in a lab flow reactor, this is still a fume hood size, we showed some very encouraging results. So here you can see how the packed bed reactor works. We have a column, like in this setup here, and in this setup, a even bigger column in an HPLC oven. This column is filled with immobilized enzyme. And what we fuel is fish oil and ethanol, and what comes out is the product, but without the membrane part in this case here. So we performed some flow experiments in smaller scale here and there. And then we applied what we have learned into a even bigger reactor like this. And then we went on and saw that from the crude fish oil, we were able to perform the enzymatic reaction. In this case, we only reached 45% conversion, but to our surprise, we saw that the membrane was able to add another selectivity so that we were able to increase the omega-3 content even further. So the selectivity here from the membrane can be explained that it also distributes between different ethyl esters or, for example, monoglycerides, and sorts out the right amounts to our favor. So what came next is to engineer a reactor in pilot plant scale, where we envisioned also a reactor in a series, 3 reactors in series, to come up with a number up of the scale up. And here you can see a reactor in real life, so to say. And this is the scaffold of the reactor. And in the end, we were able to show that in a 3 years amount of time, we were able to come from just an idea to a lab-scaled pilot plant. And we were already successful implementing the reactor at our site here at Enzymicals. This pilot plant has a throughput of 2 to 5 liters per hour with immobilized enzyme in a packed bed reactor. And the first results were promising and encouraging. And we are now working on optimizing the process even further. So stay tuned for updates. With this slide, I want to end my presentation. So I was showing you that at the beginning of the project, in such a huge consortium, we were able to accelerate the process of the developments. And after 6 months, we were already doing the basic experiments, and we achieved technical readiness level 2, so to say, after a half year. After 18 months, we validated the concept at the technical readiness level of 3. Then at 30 months, TL4 was completed with this experimental pilot setup that we had in place. And now at the very moment, we are in 42 months in project time. We are at technical readiness level 5. At the moment, we are demonstrating the process successfully, and it is foreseen in 6 months that the industrial pilot plant will be implemented at our project partners Solutex in Spain. Yeah, stay tuned. You can get updates on our project progress on macbeth-project.eu. You can find us on LinkedIn. And with that, I want to thank you for your attention from the Bock case. Thank you very much.
Thank you very much, and thank you for sharing this exciting story with us. I have a question here from Wilhelm Huck. What is the lifetime of such membranes in the packed bed reactor? Does it need any measures to keep them working or regenerate them?
So this is an interesting question, because it's not only crucial to have the membrane working for a longer time, but also the enzyme. And I was rather expecting this question, but I can also elaborate on the membrane part. So we have results from a half-year period now. When we had the first reaction mixture in place, we sent some material to our partners to the membrane part. And for 6 months, we see that the membrane keeps on performing. And unfortunately, long-term results will only be part of the next 9-month project time to gather long-term results also on the membrane longevity to keep them running. So unfortunately, I cannot answer this question yet in full length.
Okay, thank you. Anyway, maybe a follow-up question, since you mentioned that. What is the lifetime of the enzyme at the moment?
So we only performed the first experiments with this setup for 2 weeks now.
All right.
But we, with another setup, I think it was on the slides as well. Yeah, this setup on the bottom here, which is just a very ordinary and simple HPLC pump into an oven device, we were able to have the enzyme be productive for 4 months straight without losing its activity.
Okay. This sounds indeed promising. Well, I don't see any other question at the moment. So thank you very much, David, for sharing these results with us. That was highly exciting. We have now, for the last 20 minutes, planned a panel discussion. And well, maybe if you stop sharing. Okay, perfect. We have planned a panel discussion whereby either people can ask questions which were not answered before, or we have some questions here on the list which we thought could be interesting for some people. Like, for instance, what are the challenges in flow or in cascades? Which breakthrough innovations would be needed to push biocatalysis in flow? Which breakthroughs, innovations would be needed to establish biocatalytic cascades in industry? How does flow perform against batch reactions, when you would recommend using what? What are the challenges in immobilization of enzymes? Is it preventing large-scale application? What is the future of biocatalytic cascades in industry? What is the future of biotransformations in flow for industrial applications? Where do you see the showcase innovations playing a role in the industry? Well, probably we cannot answer all of those in these 20 minutes, otherwise we would stay here too long. But maybe if one of the panelists would like to contribute to one of the questions, or choose one and give an answer to that one. Well, I can give it a try. David, you were just last. When you would compare flow against batch reaction, when would you recommend to use what, or is there any possibility there?
Yeah, I think there are 2 measures that you have to compare. So first of all, it comes down to the catalyst. So, and there are 2 questions to answer. First of all, is the catalyst cheap or is it expensive? If it's cheap, you will opt for batch processes, since they are there since centuries, they are highly optimized, and flow always brings some novelty in place. So, but when the enzyme becomes expensive, then you want to have it regenerated in some case, or use it in a flow process. And when you want to apply a catalyst in a flow process, you have to look at what we talked about very recently, the longevity, and in this case, the enzyme, which is a very crucial part, and it's really not that easy. I just said, yeah, it was alive for 4 months, but it took us 3 years to figure out how we kept it alive for such a long time. And this is very crucial. You cannot have a flow process with an enzyme that is only living for 10 minutes or half an hour, or you may have to make the reaction conditions in such a way that it's favorable to the enzyme to be alive. And this is where CapEx is a huge question, because when you implement a flow process, you need to have a tailor-made reactor instead of a big tank, which is already there. And this tank needs to run. And if it's just a slight improvement, no one will opt for flow. You have to make big advantages in flow compared to batch in order to initiate the process to go from batch to flow. I think these are my
Thank you very much.
summary.
Yes. So it was cost, stability. Any other panelists, any comments to this question?
Yes, if I can say something.
Well,
So I fully agree with David, of course. So this is reality. And I guess it depends a bit on what type of process you are developing. So we have been developing bulk processes with a single enzyme, where the goal was keep the enzyme alive as long as possible. And if it expires, can you recycle the carrier so that you get the cheapest bulk process you can imagine. At the same time, if you look at new API processes, where sometimes 10 different enzymes replace a completely chemical route, it's clear you have to focus on multi-enzyme immobilization to put the enzymes together on a bead. There's a nice article where a cofactor recycling enzyme and keto reductase are combined on one bead. And you can see that the catalytic amount of NADPH that you need to add can be reduced with a factor of 100. And so this is going towards synthetic biology. And we don't only have customers who ask for 2 enzymes put together on a bead. We have those who want to do 5 or even 35. And so I think in the next decades, we can see that this whole system should replace a living cell, and then everything is concentrated on a carrier, and either in a batch or in a column. So this is what is desirable, and we are slowly working towards that goal.
Thank you very much for this contribution. Anybody who wants to add to this question? Some comments? Suggestions? Maybe there's any suggestion to another and a different question you would choose? Or do you have a burning comment? Matthias? When
Yeah, I lost the questions because they're not anymore up. But I think there was a question also regarding what could be the future for cascading industry. I think we have very good, thanks. And I think we have discussed today lengthily about it, and I think we all believe here that there is a bright future for biocatalytic cascading industry, and we maybe just see recently we also have seen maybe some key publications that have basically brought this again, and I think there is a bright future, but there are also still some challenges in order to deploy the full potential. And I think that as we discussed one today, for sure the immobilization of a lot of enzymes in the same carrier is one, but as I also said in my talk, the optimization of such system is still very complicated. So in order to really develop full potential at scale, I think this is for sure. And then we didn't talk today a lot about enzyme engineering, but also this in order, to improve enzyme to such a level that then they can be used in industrial scale for natural reactions and so on. So I think there is a lot still to do, and it's slowly, we are slowly moving, but I think there are still quite some challenges in the future.
Thank you, Wilhelm.
Yeah, I was intrigued by earlier comments from David, who quite clearly mentioned that if your sort of flow process doesn't really make a big impact, then nobody's going to implement it. But then Rob said that people are nowadays looking to immobilize sort of multiple enzymes, up to 30 enzymes in one go. Then I thought sort of many of the questions that we discussed on optimization really become a major issue, right? So I can imagine that if you have 35 different enzymes, how do you sort of tackle this problem of optimization? Because it's going to be a very expensive sort of process and laborious process if you do this sort of one enzyme at a time. Is this something that people are already doing, or do you say, well, at the moment that is wishful thinking, that's where the field is going towards?
I would say this is in the hands of those companies who develop new enzymes. So basically, it's the desire to do metabolic pathway engineering, but then have all the enzymes separate and only those ones that you want. And I guess there are many scientists in this world that say, well, why take all the guts out and try to do that in a laboratory? Why not just keep the bug alive and do everything with fermentation? And because I think what we all should realize, our biggest competitor here, or friend, whatever you name it, is fermentation. So isolating an enzyme is always more work than just doing fermentation. And if you can ferment a simple starting material into a complex molecule, and then, well, it's not synthetic biology, it's just living biology, but you also get to the goal. But you can imagine it will merge somewhere, it will meet in the middle.
Mm-hmm.
Maybe I can ask a question there to you, since you are here the specialist for enzymes in applications. Rob just mentioned isolation of enzymes. When you immobilize, what is the preferred way? Is it to have purified enzyme, or is it also feasible to go with crude extracts, or even just take the cells itself and immobilize that? Are there any experiences?
Oh, yes. Yeah, so usually we prefer to use cell-free extract that contains all the proteins, because for some reason it always seems that the enzyme that you want to use is more stable if these other proteins are present. They should be not present in such a level that they interfere with the immobilization capacity, and then they can sit in the way of the enzyme you want to immobilize. But usually highly purified is not necessary. But if you go to affinity immobilization, which is used for multi-enzyme systems, because then you have the problem, if you have two different enzymes, how can you make sure that they immobilize with the same speed and that one does not push away the other? This happens with classic immobilization, where adsorption takes place, one is more hydrophobic than the other. But with His-Tech affinities, you can have equal shares of enzymes immobilized. And if you use such a technique, then basically you will always work with the most pure enzymes that you can imagine, because all the other proteins don't have the His-Tech. And in that case, it usually works quite well, and you see activity recoveries of up to 100%.
Okay, thanks for this statement. Any other comments to that? But when you would have now, just to follow up, Rob, well, with the His-Tech, it's clear, but when you have it now multi-enzymes and would immobilize that, so you would go for a different tags, or which technique would you go use then there?
Well, basically, if they all have the same tag, they should all have the same affinity strength.
Okay. All
and
right.
then if you mix the enzymes in the right proportion, and so I gave the example of the ketoreductase and the cofactor recycling enzyme. Usually it was best to immobilize them on separate beads because you could not optimize the immobilization for two enzymes at the same time. And you should do the two differently and maybe even on different types of beads. But if you do it on a his-tag, and then you should have a bead that does not absorb the enzyme as well. So there should be no interaction. That's why very hydrophilic beads like agarose or cellulose are interesting because they do not have a lot of interaction with the enzyme. And then you get equal binding. But with that example of ketoreductase and the cofactor recycling enzyme, only when you're using his-tag, you can immobilize both on the same bead, and then you can use the lowest amount of catalytic amount of cofactor that is then endlessly recycled. With any other approach, it does not work that efficiently.
Right. I mean, maybe some of the panelists has a question as well in between, or because I have a follow-up question. I mean, now it seemed from the answer now that the his-tag is a very well suitable tool. How does it compare with other options for immobilization? Well, a good question maybe for the enzyme to go there, or also for insight there to theory. Anybody wants to answer that?
I can give my opinion. Yeah.
Right. Yes, that's what it is about.
Yeah. Just one comment also about to add on what Rob had said before. When trying to immobilize more than one enzyme in the same bead, we have also tried to use specific chemistries for each enzyme, and that seemed to work to some extent. Obviously, when you're using affinity tags, it works quite well because you have the same rate of immobilization, and then depending on the support, you can play a bit. He explained it very, very well. In terms of the his tag compared to other immobilizations, we have seen that the immobilization through his tag normally gives you a high recovered activity depending on the material, if you can avoid those interactions. But then it's more a problem of the stability of the enzyme. How much do you actually stabilize the enzyme? For example, if you want to use it at higher temperatures compared to the covalent binding. That's what I can say in our experience.
Okay. Other comments? Well, we during when we started the discussion, there were the comments why is sometimes flow preferred. Well, there are clear parameters. When there would be the wish list, what should be the next project funded on? So which research is needed to push it forward, be it flow, be it batch? What would be a general topic there? What would you suggest? What is the urgent need? I mean, we had obviously the obvious answer could be make enzymes cheaper, and the other make it more stable. So these are the obvious answers, and improve co-immobilization. So that's parts of that. Is there anything else you would put up there?
I think in general, a nice industrial application where you can use enzymes, where enzymes have maybe not been used before, can really serve as an example how versatile the technology is. And obviously it needs to be high end, so not something that you could have done, say, in the 70s, but something that you can do because you have all the technologies available right now. But I think there's always a cost factor, and that is the development cost itself. So if you look back at the sitagliptin enzyme, so Merck spent like 25 million on developing that single enzyme. I think nowadays that could be done for much less, but imagine a world where you could do this for, say, a few 100,000 euros and in a few months' time. then yeah, the technology will have more impact because you can apply it more. So you made a small joke, make it cheaper, but I think if the technology, the state-of-the-art technology is, if it becomes common day technology, this is where you see the breakthroughs, because then it is used for any thinkable application.
Thank you. Stefan?
Yeah, so I agree with everything said. One additional thing that might be important for getting more biocatalytic processes, especially immobilized enzymes, into industry would be making the developments faster and more streamlined, because as we heard, there's this need for speed, so it can never be fast enough. So everything that helps shortcut development times, have more insight from data that we gain from experiments. So everything that goes into this direction, I think, will be very valuable in the future.
Perfect. I think this is a good point to say that we all together keep on pushing the flow and pushing the enzymes either in cascades or batch, never mind, to have more showcases from which we can then learn and even improve further. So I think with this statement here, I would close the workshop and thank all speakers from the various companies for participating and for their presentation. I really enjoyed the afternoon, and well, in case any questions or contacts, so you've seen who is doing what. I hope there will be some other additional contacts and exchanges. So thank you very much for participating.